So within the group of plants that we call the angiosperms, which were the broadleaf plants, flowering plants, we typically have two groups. Traditionally, they were just called monocots and dicots, but the dicots have been split into two groups, and most of them reside in this group now referred to as the or true dicots. So we have still monocots and then the eudicots. And so what we're going to look at are just a quick list of the characteristics. I'll talk about a couple of them, define a couple things if there's something you don't know. We'll look at the cross-section of a stem briefly. You'll probably see better pictures in the book with some labels, but I'm just going to kind of orient you to some of these. And then lastly, I'll talk a little bit about, within the dicots, secondary growth as a little bit of a topic, and then that's kind of where we'll be ending. So, two groups, monocots and dicots. The eudicots are often referred to as sort of the woody plants because they are the group that can produce wood. Now, something to keep in mind as well later as we get to talk about secondary growth, that the gymnosperms can do this as well, and gymnosperms can produce wood. So this is not the only group that is wood producing, but it's just that within the angiosperms there's a group called the monocots. It has about 75,000 species in that group. Pretty sure, something like that. 75,000 species. And that's an estimate. They don't produce any true wood. Some of them, like bamboo, produce products that have wood-like properties, right? But technically not wood because of the xylem, the way in which it grows, which I'll talk a little bit about here. So get into the characteristics quick. So the name cot, all right, refers to a structure called a cotyledon, which is sort of an embryo leaf. Right? So it's a structure found in the developing embryo, and the monocots, mono has one, and then the dicots have two. So that's kind of like the named feature of difference between them, although there are then a whole number of other differences, which we'll sort of talk about. The leaves of monocots, all right, tend to be more like blades of grass, for example. So grass is the most common type of monocot, or at least the most familiar one to most people. All right. And the veins in that, we talked a little bit about veins in the previous lecture, are parallel. So the veins run throughout the blade of the leaf, kind of right along with one another like this. Whereas the leaves of eudicots have net-like veins. So if we had a leaf structure, they branch, and then branch, and then branch, and then branch again, and so forth. So we got to get those branching structures. So versus the two types. Flower parts. So previously we looked at different parts of a flower. Typically for things like, well, the easiest one to usually count are petals. All right. So if you look at a flower and it has 6 petals that you count like this, then it is most likely a monocot flower. And then if you look at what do the leaves look like, and you see, oh yeah, they have parallel veins, you won't be able to see the cotyledon unless you were to look at it. We will be able to look at a cross section potentially, so that's something we could do. So some of these things you'd have to look under the microscope. What could you actually see by looking at the plant? Probably can see the leaves, and you could probably see a flower if it happens to be flowering, and then you can use these characteristics to decide, is it a monocot or a dicot. For the flower pots in the eudicots, it's a little more complicated in that we have multiples of 4 or 5. So you may just have 5 flower petals versus 6, so they go, okay. Or you could have multiples, so like there's 8. So that would be a multiple of 4. So that's the type of thing. Now, then there's compound flowers and things that make it difficult to count flowers that have many, 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 many petals on them. But for sort of wild flowers, not ones that have been bred, these are the typical numbers that you're going to see. So now we'll get to the vascular tissue. That's what these pictures are relating to. So this is a cross-section of, cross-section, we usually refer to it like XC for cross-section, and this is monocot stem. And this would be a cross-section of a dicot stem. And it's not as detailed as the pictures that you would find in your book, actual photographs of the cross sections. But I'll be able to talk about some of the basics. So on the outside, you obviously you're going to have a cuticle on both, followed by an epidermis. Okay, so first a cuticle, then an epidermis. Then you might see a cortex, most likely. And then what we're really focusing on here, though, you have a sort of pith tissue in here, ground tissue, and other things that we're not going to be very concerned about from this part of it. It's more maybe in the lab section. If you're looking at slides and comparing and naming them, you'll kind of look at the pictures and point to which area has which characteristic or structure. What we're looking at really, in this case, are the vascular bundles. And so I'm going to grab a different color just because the orange for this one probably isn't going to work as well. Should grab something else. Okay, so all these structures here that you see kind of going around here are the vascular bundles. Now, the vascular bundles here, are like these structures here. So you can see there's another one over here, this one here. So they're all scattered around, and that's kind of the characteristic here. In the monocots, the vascular bundles are scattered. Vascular bundles. Whereas here in the dicot, the vascular bundles are in a ring-like structure, which is going to be important in a second as we talk about secondary growth. And so it's very characteristic if you were to look at a cross-section of the leaves where you could see them. So what you have is you have your, like you see a vascular bundle like this, you would have some xylem to the interior. Then on top of that, you would have the phloem. Okay. And then this would be more the supporting tissue like a sclerenchyma. And each bundle is pretty much the same, kind of repeats and goes around in a ring. And they have the xylem and the phloem as well, and other tissues here within these little vascular bundles, but they look a little bit different. The arrangement is a little bit different. They're a little more simple in structure, and then again, they're not organized, they're more scattered. So monocot versus dicot, cross-section, pretty easy to tell because of the ring structure, and it'll be easy to remember in a second as we talk about the next thing. So we're talking about wood or secondary growth. So we started off saying that the dicots were the woody plants. We're not going to get into a lot of detail in this, but we'll bring this up. Okay, so we're going to have what's referred to as primary growth, which all plants are going to have. So for all plants. And then there's secondary growth. That's not for all plants, and that's what I'm going to talk about here. So primary growth is our growth of our roots, stems, leaves, branches, flowers, etc. Okay, growing from meristems, often apical meristems, the tips of the shoots or tips of the roots where rapid cell division takes place and the plant grows longer. And then can give rise to these other structures. Right? So there could be ones that branch out as well, and then they are also a type of meristem. Now for the dicots, okay, the ones that have this vascular bundle arranged in a ring, what we have is, does it work for us? Yes. Okay. Try to line it up as best as I can. It's not going to be perfect. Okay. What will eventually happen with most of the dicots is that over time as they continue to grow taller, they'll also start to grow wider. All right. So this is secondary growth, is growth in width. the plant gets bigger, and it's going to be the result of really two different types of meristems that are lateral meristems to grow sideways. Right? And one of the primary ones is called a vascular cambium. So the vascular cambium is a ring that forms kind of between the xylem and the phloem of this plant in the stem here. And it can happen in the roots as well. So it's a structure that will form in these areas to allow the roots to grow wider and the stems, to grow wider as well. What will happen is in the vascular cambium, there'll be cell division, and then cells will move, right, to sort of maybe partly toward the outside and then some toward the inside. The cells that move toward the inside, so the cells that move to the inside will become more xylem cells, but now in addition to the primary xylem, these are secondary xylem. Which is really what's going to become our wood, right, later on. And then the cells that move to the outside of this ring structure, so like in this direction, will become secondary phloem. Right, so it's a somewhat complex topic, and there's a lot more to it than this, but I'm just going to give you the basics and a few of these terms. So what will happen is essentially that will create this growth ring where the new vascular tissue will form, and it will start to then push the tissue outward. Because of that pressure, right, the outer cuticle and all has to resist it, and it probably can't, and it's going to kind of burst. And so we have another cambium, another sort of meristematic ring closer to the outside. So we have an outer meristematic ring called the cork cambium. Okay. So we have a vascular cambium, which is creating new vascular tissue and laying down these growth rings. And then you have a cork cambium, it's more toward the outside, and it's also going to do the same sort of thing, but produce different types of cells that are going to form cork, which is going to then protect the outside of the plant stem as it grows. And then this will continue to lay down and become thicker over time, and every time the plant goes through a cycle of growth, it'll add to that and grow. Again, I said this can happen. It will happen year after year when you see talk about growth rings on, say, a tree is cut down, and you cut right through it, and you look at it, and people count the rings to see how many years old the tree is. Typically, this is the idea behind what they're counting, is that this ring of tissue kind of grows rapidly during the growing season, lays down new tissue, and then in a dormant cycle of the winter, it really slows down and pretty much stops the growth, and then it starts up again. And so then you see these patterns of more rapid growth and really slow growth, and it lays down different types of the xylem so that you can kind of see bigger or smaller tubes, and you kind of can see that pattern, the ring patterns. And you can kind of count growth cycles for the plant, which are typically year cycles, and so you can count the years of the plant. So, like I said, there are a lot more to this, but the main terms to take away from it are really the vascular cambium is this ring of growth that happens right here through the vascular bundles, right, that kind of links them together, and then that's where the growth takes place in that ring, and then that continues to happen year after year after year, and it keeps kind of pushing the outward and then kind of building the inner part. And there's all different kinds of cells and tissues there, but that's the basic idea. That's only happening here in the Eudicots, or the dicots that are the woody plants. It does not happen in the monocots. The monocots don't, because they're scattered, they don't really form a ring structure like that, and so you don't see them. Typically, they are then just on a sort of a yearly cycle. They're often referred to as annuals, so that the plant often doesn't survive a winter. And so maybe the seeds will survive, and then they'll go into the ground, and then new plants will come up, but the original plant dies off over the winter time. That's generally what happens. Last couple differences then have to do with the seed, and the pollen, and how to tell the difference between them, which again you need a microscope to really see. You wouldn't just be able to look at the plant and tell. So if you were to look at the pollen of a monocot, it has one little opening in it, whereas the dicot pollen has these 3 little openings. They're called apertures, but they're little openings in the pollen. In addition to that, if you were to look at the seed, the endospore tissue is that tissue that then grows around, so the 3N, if you remember, tissue that grows around the embryo to provide nutrients for it. The embryo consumes those nutrients and the endospore gets all used up in the dicots, so at the end of development. But at the mature seed, there's still endospore present for the monocots, which can potentially provide them additional nutrients going forward. Whereas this is all used up, but they're going to have to find the new nutrients from the soil. And so that's kind of the main difference here. So you should kind of know what some of the differences, be able to list about 5 differences between monocots and dicots and explain each of these differences. And that's kind of what we're going through here. As far as the cross-section goes, you're not really responsible for every single thing in cell type, in here. Just kind of the basics, the identifying the patterns of the vascular bundles for the dicots versus the monocots, and then how the pattern in the dicots is related to then secondary growth, which is the woody growth, so the lateral growth of the plant as it grows wider, and whereas primary growth is growth from the end meristems where it's growing taller. And that's kind of pretty much it. We'll kind of do one more sort of some summary of plants to kind of do some comparisons between the non-vascular plants, the vascular plants that do not produce seeds, like the ferns, and then the vascular plants that do produce seeds, the gymnosperms and the angiosperms, and kind of just do a final comparative summary, and that will be the end of the plant material.